Hello everyone. We'll start with the chapter of transport in plants, that is chapter number 11. Now, as you know, because of this lockdown, it will be impossible or to schedule the lectures. So I'll be taking lectures in this form. I'll be uploading it to my YouTube channel Naman Trivedi Biology for NEET. And to view this type of screen recorded lectures, please download it and then view it, because right now, as per the YouTube notification, there has been lot of overuse of the YouTube servers and they are under too much load. So YouTube has issued advisory that none of the videos will be bigger than 240 pixel in size. So even though I'll upload the best quality, you'll be able to view it in an inferior quality. So if when you'll click it, when you'll stream it on YouTube, you won't get the clear images. But for understanding with respect to my other videos and from the suggestions received from my other students, this is a better way. So please download the video and then watch it, and then mainly put your headphones and then listen to it. As you don't have too much options, and just to make it easier, I'm using the same textbook thing. Okay, I'll be underlining, I'll be saying which part is important. So we'll start with the chapter number 11. Okay, and then I'll take chapter number 12 in the subsequent series. Now also, if you are interested, you can Telegram, make a group in Telegram. I'll send the video lectures in Telegram also, because then you'll get a clear image. Okay, so do not forget to download and then view. So first subscribe my channel, then download it, and then you can view it. So we'll start with this chapter, that is transport in plants. Today we'll cover the first part of the chapter. Now, as you know that plants need to move the substances at very large distances, okay, and they have a huge body structure, okay, as compared to the animals. Now plants does not have any of the circulatory system, so they have something called what we call as the roots, which will absorb the water and minerals, and then it will reach to other parts of the plant body. Then photosynthesis will mainly occur in the parts which are green. Okay, so green parts will mainly synthesize the food, or they will form something termed as photosynthetes. So it will be formed in leaves, and then it has to be transferred across the plant body to the other parts. So as we know that certain substances from the root will move towards the upper side, whereas certain substances from the leaves will move towards the roots. So photosynthetes will move from leaf to the root, while water and minerals will move from root to the leaf. Okay, so I hope you know that thing. Okay, so that is what they're trying to explain in the first paragraph. We have also covered this part in our regular lecture, if you remember the last lecture which had happened before the corona situation. So how these substances will move? So there are some of the transport processes, like if you know about diffusion, okay, which we have discussed in cell membrane also. So it is the movement of a substance from higher concentration to lower concentration. That is what we call as the diffusion. Now over here they have given the most important part. In this part they have mentioned the definition of the translocation. Okay, so for small distance, plants can use diffusion. If it is to be moved inside the cell, the plants can use cytoplasmic streaming, that is the circular movement of the cytoplasm, or what we call a cyclosis, and also they can use active transport. For large distance movement, it will mainly occur through vascular system. If you know about chapter number 5 and 6, we have that has been mentioned in the 6th chapter in plant anatomy chapter, that vascular system is mainly consisting of xylem and phloem. Vascular system will mainly consist of xylem and phloem, and this xylem and phloem helps role for the translocation. So over long distances will proceed through vascular system, which is mainly by the vessels like xylem and phloem, and such a transport is termed as translocation. So if a substance is moved from root to the leaf of the same plant, which is say 10 meter 20 meters in total height, then that is mainly through translocation. Okay, so just go through this paragraph. From this paragraph, for questions, translocation is important. Coming on to the question from this chapter, they may ask you 1 or 2 questions, okay, or up to 3 questions may be asked. But the prior 10 chapters which I have taught you, they are more important in comparison to this chapter. It's a very interesting chapter. Now, if we discuss about the direction of the transport, then in rooted plants, the xylem transport, that is xylem is responsible for water and mineral transport. It is unidirectional, that is, it will always happen from root to stem. However, the organic and mineral nutrients can undergo multidirectional transport. Okay, so organic compounds, as we know that they are the organic compounds are mainly formed by leaves, which where there is chloroplast present. They perform photosynthesis and then they'll export it to all the other parts of the plant, including the storage organs. Now storage organs, as you know, tubers, okay, like we have studied the different modifications in the form of the potato, sweet potato, yam and all, okay, in plant morphology chapter. So from the storage organs it can be re-exported. So from the storage organs it can be re-exported. So kindly, this sentence is important and kindly note that down. Okay, as you know, the mineral will be taken up by roots, transported upward towards the stem, whereas, just a moment, whereas leaves, okay, and will mainly form the food which will be transported downwards toward the roots and other parts of the plant body. Then they have mentioned that whenever any plant parts undergoes the senescence, the nutrients will be withdrawn from such regions. Okay, also if you see, then hormones or plant growth regulator, the 15th chapter is all about this. They are formed in very small amounts, but they can be transported in unidirectional manner from the place of synthesis to other plants. So you can correlate this transport to the endocrine system, which you have in the humans and animals. Okay, so there are different ways of transporting different substances. So just go through this paragraph. Now coming back to the means of transport. So we have diffusion, facilitated diffusion, okay. Then we'll discuss about the other parts, as you know, active transport, and about the their comparison between them. Okay, so right now we'll discuss mainly 3 modes, that is diffusion, active transport and facilitated diffusion. Now as you know, diffusion is always from higher concentration to lower concentration. That is the movement of anything which is for short distance from cell to cell or from the intercellular space of the leaf to the outside, but it is always in random fashion and it will always move from the higher concentration to lower concentration, as you have discussed this also in the chapter of the cell. So it's a slow process, it is not dependent on living system. So it's a slow process, it is not dependent on living system. It's basically based on the principles of, just a moment, excuse me, it is basically based on the principles of chemistry and physics, as you know. Okay, so it is not dependent on living system, as you know. You should note that down. As you know, the gases can diffuse very fastly, while liquids will be the second best and the solids will be last one, or they can rather not diffuse. Okay, diffusion is very important for gaseous movement in the plants. Okay, now the rate of diffusion can be affected by 4 things. That is by gradient of concentration, the permeability of membrane which separates it, and the temperature and the pressure. So you should know there are four things for this, that is the gradient of concentration, the permeability of membrane, as well as the temperature and the pressure, okay, which will between the two mediums. So this is what we call as diffusion. Now next part is about a type of diffusion which we call as facilitated diffusion. A type of diffusion which we call as facilitated diffusion. So this mainly represents that there must be gradient must be there. So first and foremost thing is it must have a concentration gradient. Okay, it is primarily thing for this sort of diffusion. Okay, CG stands for concentration gradient. Then there is difference between the diffusion rate for different substances, because substances can be smaller, can be bigger. So smaller will have larger or higher rate of diffusion than the bigger or larger substances. Okay, then substances soluble in lipid can diffuse faster. As you know, the cell membrane contains more lipid, so hydrophobic compounds can diffuse faster, while the substances which are, let's say, hydrophilic or which has more water, they will take more time to diffuse, or their movement cannot occur directly. So such movement, so the movement of these hydrophilic compounds is to be facilitated. So the movement of the hydrophilic compounds is to be facilitated. So if you want to move the substance which is hydrophilic moiety, then their movement needs to be facilitated, and for that we need facilitated diffusion. Now how this movement will be facilitated? So for their movement there must be a concentration gradient, because remember it will be always along the concentration gradient, and it will be facilitated by what we call as proteins. So there will be some proteins which can play a role in this facilitated diffusion. These are specialized proteins which without use of ATP can transport molecules from a concentration which will be high to low concentration. Okay, so remember that. So it is always from, the movement will be always from high concentration to low concentration, or what we call as along the concentration gradient, or what we call as along the concentration gradient. ATP is not used, okay, and it can never occur from low to high. Now when the transport rate of this will reach maximum when all the protein transporters are used, that is when the entire membrane and all its facilitated proteins are saturated. When the entire membrane and all its facilitated proteins are saturated, then only the transport rate will become maximum. Okay, now this facilitated diffusion is highly specific. Okay, it is also sensitive to certain what we call as inhibitor. These are the compound which can inhibit the facilitated diffusion. These are the compounds which can inhibit the facilitated diffusion. Now they have given examples of some of the proteins. They have given examples of some of the proteins which are like porins. So this protein can form channels, okay, in the membrane, and these channels can be open, can be closed, and if they are open they will allow the movement of the certain substances. So porins are nothing but pore forming proteins. The porins are nothing but pore forming proteins, that they form large pore in the outer membrane of plastid, mitochondria and some bacteria, and will allow some of these substances which are smaller in size to pass through it, some of the substances which are smaller in size to pass through it. So such pore forming proteins are termed as porins. I can also give you one more example of porins, that is of the nucleoporins, which is found in the nuclear membrane. Okay, now they have given the example of something termed as aquaporins. So aqua means water, and there are 8 different types of aquaporins which can help in transport. Okay, so what they do, like they'll take a protein, they'll rotate and release the molecule inside the cell. Okay, so now try to understand this thing. Okay, I'll discuss the diagram number 11.1. So if you look at this diagram, this is our plasma membrane. You can see this part is mainly lipid, and this part, the pink part which you see, it is our protein. This dark red line which I am doing is our protein. So these are the substances which are outside the cell. These are the substances which are outside the cells. Now they have to be moved onto the inside. They have to be moved onto the inside. So for that movement, as you can see, they are moving towards this pink part. They are moving towards this pink part. Okay, in this direction. This is what I am, the thing which I am repeating again is the direction in which they are moving. So how they will move? So they have to be moved along this pink part which you can see. They have to be moved along with this, just a moment, yeah, with this pink part which you can see, or this green line I am doing. So they are supposed to pass through this part. You can also see the arrow over here, So if you look at this structure, of these proteins, then they are something like this. So a thing can move through this part, will pass through it. So it works as a channel. Okay, now how they will function? So if you want to understand that, you should know this diagram. Okay, see, so they'll take a substance from here, it is rotating, and then it is releasing these compounds onto this side. It is releasing this compound, as you can see, onto this side. So the substance which were here have reached to this side. How they have reached? They have reached through passing through these proteins. And this protein, this pink colored compound, what you can see, it is our facilitated protein, or it is sort of porins which form the channels. Now remember one thing, all the proteins cannot do this sort of rotational movement. Only some proteins can do it. So if you remember, there was a question, okay, about the flip-flop movement in the 10th chapter. Now that question has been asked 3 times in different ways. So always remember, usually lipids will show quasi-fluidity or lateral movement. Proteins can also show lateral movement, but some of the proteins can show this kind of rotational movement. Not all will show this. Kind of movement. Keep that in mind. Okay, only some of this. So if they ask you this question, your answer must be aquaporins can show this kind of movement. Okay, so proteins can exhibit lateral movement, can exhibit translocational movement, and can also exhibit rotational movement, but only to some extent. So rotational means it will completely get oriented in opposite direction, उल्टा हो जाएगा और फिर रिलीज करेगा. That is what they are trying to explain. So यहाँ से ये जो part है, as you can, just a moment, ये जो part है यहाँ से it will take. Okay, so if this part is like this, okay, this part will again, it will change its orientation, and so that the thing which is over here can be released from this part. But not all proteins can exhibit this. Be careful. So from facilitated diffusion, remember this example of porins and aquaporins, and this diagram, it is highly important. 11.1 in this aquaporin part. Now coming on to the passive symport and antiport. Now this facilitated diffusion can be of three types. It can be of three types. Okay, so as you can see, it can be symport, it can be antiport, it can be uniport. So if only carrier protein will allow the transport in one particular direction, if only a protein will allow a transport of single molecule in one particular direction, we call it as a uniport. Uni means one. If it allows transport of two molecules, if it allows transport of two molecules but in opposite direction, we call it as a antiport. And if it allows transport of two molecules in same direction, and if it allows transport of two molecules in same direction, we call it symport. So the first one is uniport, second is antiport, and third one is symport. Now you should know that in symport both can combinedly cross in same direction, in antiport they move in the opposite direction, while in uniport only single will be moving across the membrane. While in uniport a single will be moving across the membrane, then only the transport is possible. So that is what about antiport, uniport and symport. Remember, the sim means together, anti means opposite, uni means one. Now we are discussing in the last part of the lecture about the active transport. So active transport mainly is based on the energy, and it is against the concentration gradient. So pumping is done against the concentration gradient. So we also have different proteins which play a role in active transport. There are some proteins which play a role in both sort of transport. Now for this purpose mainly we have pump. If you remember ionic pumps which we have discussed in the nervous system, or now impulse conduction. So these pumps are the proteins which can use energy to carry substances across the membrane. So these pumps are the proteins which can use energy to carry which will lead energy to carry substances across the membrane. They can transport from low concentration to high concentration. That is, it is what we call as against the concentration gradient. It is against the concentration gradient. Okay, as you know, it has been mentioned in this part. And such transport is also termed as uphill transport. Over here also maximum transport can be reached when all are being used, when all the protein transporters are used, and they are also sensitive to inhibitors. Okay, and they are very specific in their role, just like enzyme substrate combination. So ATP is used and it is against the concentration gradient for the active transport. I hope you know that. Now this is a very important table which you should know, where they have basically used certain criteria, like membrane proteins are required, yeah, in facilitated active required. A facilitated active is highly selective, saturation can be achieved in both, yeah. Uphill transfer only in active, ATP only in active, rest of all over here it is not required. Simple diffusion, no proteins required, not selective, not saturated, not uphill, no, there is no need of also energy, as you can observe over here. Okay, so it's a separate thing. Okay, so this is about the first part of the chapter and we end it. So you should know about active transport, diffusion, facilitated diffusion, aquaporins, uniport, symport, antiport. Okay, and you should know the definitions, and this table number 11.1 is highly important, so kindly go through it. It can be asked here. Also remember about the rotational movement. Do not forget to download the lecture. Do not forget to download the lecture and then view it. If you have any doubts, send the question in WhatsApp group. If you find this downloading difficult, then you can use my Telegram channel. You can make a group in Telegram and I'll send the lectures. Tomorrow I will discuss with the plant water relations. Thank you guys. Naman Trivedi here signing off.